Thank you for inviting me and give me this opportunity to talk here. Today, I changed my topic a little bit because I want to cover two topics. One is about chromatin and one is about chromosome structure. I don't think I need to explain that gene regulation is a very crucial process and it's a very complex process, and this regulation happens at different levels. And today's talk, we're going to look at two different scales. One is the nucleosome, kind of the chromatin scale, and then we're going to look at the 3D chromosome scale. I don't think I need too much introduction here. Everybody here knows that eukaryotic DNA is wrapped into nucleosome, which covers 147 base pair of DNA wrapping around a histone core. Histone is super positively charged and DNA is negative charge. So together they form this very stable structure. And because of the structure is very stable, it presents a barrier for other factors to access DNA. So one example here is this NF1 is a transcription factor. And based on this gel shift, you can see this factor readily binds to naked DNA contained as recognition site. But when the same DNA is wrapped around into a nucleosome, you stop seeing that gel shift. It means this factor can no longer bind, at least within this concentration range. And that poses a huge problem for the cell, because for transcription to happen, the first step is for transcription factor to bind. So if nucleosome becomes a barrier, how does the cell deal with this problem? When you map the nucleosome distribution inside the cells, you immediately can see how this problem is solved. So this is a random segment of yeast genome that I pulled out. These arrows are open reading frames, and in between is the intergenic region. This is the measured nucleosome occupancy on the stretch of DNA. These little glitches, each one represents a nucleosome, and so this range here represents a well-positioned nucleosome array. But you can see they are interrupted by these downward spikes representing regions that has very low nucleosome occupancy. And we call these NDR Nucleosome-Depleted-Region. If you compare this map to the gene annotation here, you can see this amazing correspondence between the NDR to the intergenic region. So that means in cells, transcription factor does not have to deal with nucleosome because the place where they're supposed to bind is already open. So that raises an interesting question. How does the cell know where to generate NDRs? How do they know where to put nucleosome and where not to put nucleosome? So people have been studying this for a while, and two things, they found two factors that contribute to the NDR formation. One is the intrinsic DNA preference, because if you look at the nucleosome structure, DNA has to bend very sharply inside nucleosome. So if you have a piece of DNA like this poly A stretch that is kind of rigid and does not like to go into nucleosome, they tend to stay outside. So this mechanism certainly is very important for dictating where the nucleosome go inside the cells, but they are not sufficient. So inside the cells, there are also these sequence-specific factors. In yeast, there's these famous factors, ABF1, REP1, and REP1. They can bind to DNA and antagonize nucleosome formation near their binding sites. So in higher eukaryotes, these factors are called pioneer factors because they are the first factors that go into the chromosome. In yeast, we would also like to call them pioneer factors, but people object to it because pioneer factor was originally defined in a developmental sense. So we call them nucleosome displacing factors, NDFs, but they are essentially the same thing. So in this talk, the NDFs and pioneer factors are used interchangeably. So these factors, their main function is to recognize their binding site even when they're embedded under the nucleosome. And they can somehow access the binding sites and through an undefined mechanism lead to nucleosome displacement. And that exposes other factor binding sites so that the real transcription factor can bind and either activate or repress gene expression. So based on this description, you can understand why these factors are very important. They are the key regulators of gene expression. And so you can imagine inside the cell, if you have certain pioneer factors, they say, okay, I'm going to open here, 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 here. So this set of the genes will be regulated. And when these pioneer factor goes, and then there's a new set of pioneer factor comes in, and then they open up a different region. So they can completely change the transcription program of the cell. So these factors often function as a master regulator that directs cell fate and cellular reprogramming. So probably many of you know that the 4 Yamanaka factor that is responsible to generate IPS, induced pluripotent stem cells. And out of the 4 factors, 3 of them are considered as pioneer factors. And mutation of these factors are often implicated in human disease. Right now we know only a handful of these factors, and most of these factors are studied in a case-by-case basis. So what my lab wants to do is to do a high throughput assay that we can screen all the transcription factor inside the cell to understand how many of them can function as pioneer factors. And once we have the list of pioneer versus non-pioneer factor, we can get to understand, what's the difference between these factors and what special properties pioneer factors have, and that will generate some mechanistic insight of how pioneer factors work. Okay. This is a wrong slide. Okay, here. Before I move on, just in case some people don't know, this is how we map nucleosome. We use this enzyme called mycoalkyl-nuclease, MNase. And MNase cuts DNA more efficiently in the open region than the nucleosome covered region. So if you have a short chromatin looks like this, after MNase digestion, you would have these kind of fragments, mononucleosome fragments that's getting enriched. And then you can use standard molecular biology method like PCR or sequencing to figure out where these fragments are. In fact, the genome-wide map, nucleosome map that I showed you before, that was generated by the MNase digestion. Okay, so for our high throughput pioneer factor study, we started off with this yeast promoter called HO promoter. This promoter is a little bit unusual that it activates by 2 activators, SY5 and SBF, and both of them are cell cycle regulated. Okay, so I showed you before that for most of the yeast promoters, they're nucleosome free, right? But this promoter is an exception. If you look at this promoter, in the upstream region, it has SY5 binding sites in the open region. And then they also have SBF binding sites embedded under the nucleosome. So during a cell cycle, what usually happens is SY5 would bind first and recruit a whole bunch of factors to actually eliminate nucleosomes so that SBF can bind and activate. Okay. So for our study, we actually use a mutated HO promoter where we mutate out the 2 SY5 binding sites so that the nucleosome here becomes constitutive across the cell cycle. And we also show that this promoter almost has no activity. So here we use the MNase to map the nucleosome distribution over this region. And you can see there are 3 humps here representing well-positioned nucleosome -5, -4, and -3. When we artificially engineer the Reb1 binding site into nucleosome -4, you can see this dramatic change in the MNase sensitivity. And in particular, nucleosome -4 is displaced. And when that happens, if we use these promoters to drive the expression of GFP, we can look at the GFP expression in single cells over time. I hope I can play this. So this is the background promoter, basically this basal promoter driving GFP. And GFP is not expressed, so you can see the cells are largely just dark. But when we use this promoter to drive GFP, if you focus on one of the cells, like this one here, you can see GFP is turned on, but it also shows cell cycle dependent activation. So Rep1 is a constitutive protein that does not have too much of an activation potential itself. And the fact that this GFP shows cell cycle regulation is because Rep1 now opens up the chromatin so that SBF gets to bind and activate the gene in a cell cycle dependent fashion. So Rep1 in every sense fits into the definition of a pioneer factor. In contrast, if we put in a mutated Rep1 site that has 2 base pair mutation, none of that happens. So this is not too much of a surprise because we already know Rep1 is the pioneer factor in yeast. But the point here is that if we don't know that, we can use this assay to identify Rep1 as a pioneer factor. Also, remember, a nucleosome covers 147 base pairs, and Rep1 binding site is like 10 base pair. And in this case, we put Rep1 site at -33, means 33 base pairs away from the dyad, the midpoint of the nucleosome. But we don't have to put it there. We can put it at different locations with different orientation, affinity, copy number, spacing, you name it, right? So in principle, we can use this assay to systematically investigate the binding site configuration, its relationship to nucleosome depletion. So of course, we don't want to do this assay one by one. We need a high throughput method. And this is how it works. So we ordered these oligo libraries. So these oligos all have the same background sequence as the nucleosome -4 here. But we designed it so that it contained different, we engineered in different binding sites. I think in the end, we designed something like 180,000 different oligos, or 18,000, I forgot. Anyways, there are a lot of oligos. And then they came to us as a pool. We ligated these oligos into a plasmid background contain the rest of the HO promoter. And that generated a plasmid library. And then we integrate into the yeast genome these plasmids at a specific genomic location through homologous recombination. So this is definitely one advantage of using yeast because this kind of genetics is easy to do. So at the end of the day, we get a large pool of yeast library. These yeast all have the same genome background, but at this one particular location, it contains whatever sequence we designed. Some of these sequences will deplete nucleosome, and some of them will retain nucleosome. So to evaluate that, we take half of the pool of these mixed yeast, and we did MNase digestion followed by amplicon sequencing. So the idea is that if you have a particular sequence that is exposed, it will be completely destroyed by MNase. So when you try to PCR like this, you won't get a lot of PCR product, and you would get low sequencing reads, right? And vice versa. For the other half of the population, we just purify the genomic DNA and do the same amplicon sequencing, and that's your total input. So basically, by looking at the sequencing counts in the MNase sample versus the genomic sample, we get an effective reading of the nucleosome occupancy on all of these sequences, all at the same time. I always joke that when you come up with a new method, if you don't have an acronym, nobody cares about it. So we call this MNase over integrated synthetic oligos, MNase-ISO. This is what the data look like. So for each of the oligo that we designed, we get these 2 reads, and we take the ratio that leads to this normalized nucleosome occupancy. If we take all these normalized occupancy and plot into a histogram, you can see a very clear bimodal distribution. Most of these counts are under this peak close to 1. That means most of the oligos we designed does not actually have any effect on the occupancy. But there is a significant fraction, about 15%, that leads to nucleosome depletion. So these are the ones that we're mostly interested in. By analyzing what kind of binding sites are in this pool versus this pool, we can divide these transcription factors into 3 categories. So group 1 factor is something we call a strong pioneer factor, in the sense that if you just put in one binding site of this factor inside the nucleosome is gone. Group 2 is weaker factors where one binding site doesn't work. You need 2 or 3 before you can see a nucleosome depletion. And group 3 factors, even though they are available inside the nucleus, they don't seem to have any effect. So now the burning question is, what's the difference between these groups? What make pioneer factor? So in this plot, every single dot here is a transcription factor. The x-axis is the protein copy number, basically the concentration of these factors. The y-axis is the binding affinity of these factors to DNA. So you can see that group 1 factor really stands out as the factor that's very highly concentrated and bind to DNA very strongly. So that points to a very kind of physical picture that indicates that these factors, just through mass action, physically compete with histones for the same DNA. So to see if that is a reasonable conclusion, we took one of these factors outside and we overexpressed the heck out of it into this range to see if we can convert that factor into a group 1 factor. And yes, it indeed worked. So this is a template that contains a single RFX1 binding site. And at the endogenous concentration, it's not able to displace this nucleosome. So you can see 3 well-positioned nucleosomes over this range. But when we overexpress RFX1, you can see a clear gap showing up. We can even do this with bacterial proteins. So this is our TET-R, Tet repressor. And when we overexpress that in yeast, it actually can displace nucleosome near its binding site. And remember, this is a bacterial protein, so it has never seen a nucleosome throughout evolution. The fact it can displace nucleosome inside the cells has some clear indication of how the mechanism of nucleosome displacement. But I'll talk about that a bit later. So for group 2 factors, it doesn't seem to be very different from group 3 in this plot. But when we analyze their the structure of their DNA binding domain, it looks like this particular structure, Zinc cluster, is significantly enriched. We still don't know how this structure is compatible with the nucleosome structure, but one thing that we notice is that this DNA binding domain, the DBD, seems to be sufficient in displacing nucleosome. So in this experiment, we take the UME6 with a group 2 factor and with the DBD at its C terminal. And this is on a template that contains 3 UME6 binding sites. If you completely get rid of UME6, of course, nothing happens. So you still have well-positioned nucleosomes. But when we supply back the UME6 DBD, you can see there's a gap opening up. So that shows, first of all, the DBD is sufficient for nucleosome displacement. Second, because we know UME6 actually interact with some nucleosome remodelers in its N terminal, that indicates that at least in this case, the interaction with remodelers is not necessary for this pioneer activity. Another thing that we found in this assay is how this nucleosome displacement is related to the position of the binding sites. So we did this scanning experiment where we literally put the binding site at every single base pair on half turn of the DNA. When we did this experiment, we thought we would see 2 things. One is that as the binding sites approaching dyad, the factor would have harder and harder time of accessing the site. Also, because DNA is a helical structure, we think that the binding sites that's exposed on the outside should have easier time working with binding sites that's facing inside. So we expect to see a 10 base pair periodicity, but we see none of that. So these are the 3 NDFs that we looked at. You can see that the mutated version of the binding site doesn't do anything to the nucleosome occupancy. And the wild type version of these the binding sites deplete nucleosome to the same extent regardless of where the binding site is located. So that is a bit perplexing. And we think this raises the possibility that these factors, instead of just invading into a well-positioned, well-structured nucleosome, they probably take advantage of these cellular events where the nucleosome structure is temporarily disrupted. So they can gain access to DNA at that moment and then prevent nucleosome from reassembly. So in cells, one major event that disrupts nucleosome is DNA replication. So here, what we did is we're looking at how CBF1 disrupts nucleosome in either cycling cells or G1 arrested cells where the DNA replication is stopped. So we found that in both cases, nucleosome can be disrupted, but in terms of the disruption rate, in the cycling cells, it seems to be faster than in the arrested cells. So that indicates that some cell cycle events, probably DNA replication, promotes the invasion of the nucleosome. Okay, so all of these experiments, of course, have implication on how these pioneer factors work, but I will first talk a little bit about some biochemistry experiment before I get to that. Because we had this interesting findings with the pioneer factors, we collaborated with Michael Poria, a single molecule biophysicist at Ohio State University, to look at a single molecule level, how these factors interact with a nucleosome template. And there are two assays that is particularly relevant here. One is called PIFE, protein-induced fluorescence enhancement. So the idea is that if you have a fluorophore kind of free in the solution, it's freely rotating, and some configurations have better fluorescence efficiency than the other ones. When you have a factor that binds very, very close to the fluorophore, for whatever reason, it stabilizes the fluorophore in a more productive state, and you will see an increase in the fluorescence. And indeed, this is the raw fluorescence data. You can see that when you add the proteins, sometimes you get this uptick that represents a binding event. As you put in more and more fluorescent proteins, you see more and more of these events. And by analyzing the time in the off state versus the on state, we can get the K on and K off of the factor. And when we care about the transcription factor binding to a nucleosome, we cannot use this assay because the DNA is already in contact with a large protein complex. But instead, we're using this single molecule FRET assay. And the idea is that if you put donor and acceptor here, usually when the nucleosome is well formed, they have high FRET efficiency. But when the DNA unwraps from the nucleosome surface, you get lower FRET. Even without factors, nucleosome goes through spontaneous unwrapping, but this process is very transient. If you have a transcription factor that binds here, it will stabilize this nucleosome in the unwrapped state. So you get longer and longer of these unwrapped states. And that's basically what you see, right? This is the lower FRET here is the bound state, and that's the unbound state. So by using this kind of plot, we can get the K on and K off of these factors on nucleosome. So now we have the capability of measuring K on and K off on both DNA and nucleosome template. And the question is, how are they different? So before I collaborated with Michael, they already did some study with some non-pioneer factors, LexA, which is a bacterial protein, and GAL4, which is a canonical eukaryotic transcription factor. So this is what they found. If they compare the on rate, the binding rate, on nucleosome versus on naked DNA, they find that on nucleosome, the on rate is lowered by 2 orders of magnitude. So that is not surprising, right? Because now the binding site is embedded and takes longer for the protein to find the binding site. What was a little bit surprising is the K off is increased by 3 orders of magnitude. That means not only the factor has a hard time binding to it, once it binds, it gets kicked off much faster, okay? So we don't fully understand how this happened, but there is an attractive model here. Is that these factors bind as a dimer. So if you are a dimer and bind to DNA, you can release one hand right? But the whole complex can still associate with that. Only in some unlikely event that both hands let go, the factor dissociate. But imagine now you bind to DNA, but you also have a nucleosome, a histone here. So if you let go of one hand, this site can be immediately reclaimed by the nucleosome. So you have a hard time rebinding again. So now you let go of this hand, the whole thing just falls apart. Okay. Because the K-on and the K-off move to different directions. So the overall KD, the effective affinity, is hugely reduced. So basically, these factors have very, very little affinity to nucleosome. When we apply similar assays to Reb1, we immediately see something very different. So this is a gel shift assay, basically a bulk measurement of the binding affinity. And you can see that if you look at here, this is the Reb1 binding to naked DNA versus on nucleosome. If you look at where the gel shift occurs, you can see there's not much difference on DNA versus nucleosome. If you quantify this gel, you get the effective KD on nucleosome and DNA. You can see they're about the same. They're certainly not orders of magnitude different. So this is average affinity. How about K-on and K-off? What we found is that similar to the other factors, the K-on of Reb1 is also reduced by 2 orders of magnitude on nucleosome. So there's nothing special there. Reb1 also have a hard time finding the binding site. But what is dramatically different is now the K-on is also reduced by 2 orders of magnitude. So once the Reb1 binds to the template, it stays on there. So usually on DNA, it dissociates within seconds. When it binds to nucleosome, it can stay on for minutes to 10 minutes. Okay. And because now the K-on and K-off changed in the same direction, the effective KD is actually not changed by much. So we call this a dissociation compensation mechanism. We also did the same measurement on CBF1, another pioneer factor. And here you can see the compensation is a little bit less than Reb1, but the overall direction is the same. CBF1 also dissociate from nucleosome slower than from DNA. Okay, so which part of the protein is responsible for the slow dissociation? If you look at CBF1 structure, it contains a unstructured N-terminal domain and a basic helix loop helix domain, which is the DNA binding domain. And then there's a leucine zipper that we think contributes to the dimerization of CBF1. So what we hope is that we can delete part of this protein that we can kind of specifically reduce its affinity to nucleosome, but not to DNA. So we can find a part that is specifically responsible for the slow dissociation. But when we delete this N-terminal domain, you can see the affinity to DNA or to nucleosome did not change much. When we delete the leucine zipper, you can see that it essentially lost its interaction with both DNA and nucleosome. So by doing this kind of deletion analysis, we're not able to specifically change the affinity to nucleosome. And the only conclusion we can make is that this large unstructured domain doesn't seem to contribute to the binding to nucleosome. Okay, so we changed our strategy a little bit by contrasting 2 proteins, CBF1 and FO4. So these 2 proteins both have this basic helix loop helix domain, and they are a close homolog. There is a FO4 structure out there, and you can see is the N-terminal bits of this basic helix that's in direct contact with the DNA. And the interesting thing is because these 2 DNA binding domains are similar, they actually bind to very similar sites. So these are the recognition sites of these 2 proteins, and you can see the most important nucleotides are exactly the same, and they differentiate based on some neighboring sequence. But interestingly, unlike CBF1 that dissociates much slower on nucleosome than on DNA, FO4 dissociation is actually a little bit accelerated on nucleosome and on DNA. We also have evidence that I'm not showing here is that only CBF1 but not FO4 function as a pioneer factor inside the cells. So now we have 2 proteins that has very similar structure, bind to very similar sites, but only one of them function as a pioneer factor. So that gives us a chance of making hybrids between these two proteins to see if we can kind of morph one protein into another. So we tried different things, and one thing that worked out is when we swapped this N-terminal bit of the basic helix. So when we swapped in the full 4 basic helix, what we see is that the binding affinity to DNA did not change, but the binding affinity to nucleosome dramatically reduced. Okay. When we swapped in the CBF1 to the full 4, we see opposite effect. So if you think about the relative affinity between nucleosome to DNA, we see a 5-fold reduction in this case. So this is quite remarkable in that we now kind of nailed down to about a 15 amino acid region that seems to be responsible for the high affinity and slow dissociation of the nucleosome. We still don't know exactly how it works. We're collaborating with a structural biologist, Song Tan, at Penn State to make crystals of these proteins complex with DNA or nucleosome, and hopefully that will give us some insights. Okay, so now, given what I said already, let's think a little bit about the mechanism. So if we just one step back, and if I tell you there's a factor that can invade into nucleosome, how would you think it can work? I think you can come up with 2 different mechanisms. One we call the active mechanism. Basically, these factors can somehow dock onto the nucleosome, probably by recruiting some downstream factors like remodelers or histone chaperones that leads to the depletion of the nucleosome. It could also be this passive mechanism that these factors actually cannot deal with nucleosome, but it can take advantage of the nucleosome dynamics inside the cells. Okay, inside cells, you can have DNA unwrapping, nucleosome translocation, histone turnover, and that disrupts the nucleosome structure, and you can have transiently exposed DNA. And then these factors can immediately bind to the DNA, preventing the nucleosome from reassembling. So a major difference between the active versus the passive model is that for the active model, this pioneer factor needs to have some kind of special structure. It needs to know how to deal with the nucleosome. It needs to be structurally compatible. But passive mechanism actually applies for any factors that as long as they can bind tightly to DNA. So if you think about the biochemistry experiment we did, that the pioneer factor here, Reb1 and CBF1, they seem to have high affinity on the nucleosome than on the DNA. So their binding affinity, the relative binding affinity on the nucleosome and on the DNA is correlated with their pioneering activity. And that certainly supports the active mechanism. But on the other hand, I also showed, from the first study that I talked about, we also show that the NDFs are not that special, right? Because we can overexpress a factor and we can turn that into NDF, and even bacterial proteins, which should not have the pioneering activity at all, can also deplete nucleosome inside the cells. So that indicates that you don't need anything special to deplete nucleosome. And I also show you some evidence, for example, that their nucleosome depletion is not sensitive to the location of the binding sites. And some cell cycle events also seem to promote the nucleosome evasion. So that indicates that nucleosome dynamics inside the cells are important for the pioneering activity. This is where we are at right now. You can read what I wrote here. Basically, we're very confused at this point. We have 2 studies, and each of them makes perfect sense, but when put together, they seem to be telling us different stories. So the only thing I can say now is probably both active and passive mechanism work to some extent in the cells. And we're right now looking into which pathway plays the more dominant role inside the cells. Okay, so time is good. All right, I'm going to switch gear now and talk about something related to the 3D chromosome. So in particular, we're going to talk about engineering chromosome conformation. So in recent years, this topic becomes very popular, largely thanks to this technique called chromosome conformation capture. Just in case some of you are not familiar with this, the basic idea is very simple. Is if you want to know conformation, basically which part of the DNA is touching which other part of the DNA, if, for example, they're bridged by these protein complexes, you can cross-link everything, okay? And then you can digest the DNA and then ligate it. And then you reverse the cross-link, and then you get these DNA out. And when you sequence across this junction, you will know, okay, this piece and this piece are physically together. And by doing this kind of assay, you get these kind of representation of the data that's called contact frequency map. Okay, so when you look at these maps, okay, for every single dot inside this map, okay, like this, so that represents a contact frequency between this location and this location on the chromosome. And the darker the red is, that means the more likely these two things are together. So if you look at these maps, the most striking thing is this triangle pattern, and each one of them is what we call a topologically associated domain. That means within the domain is more likely, the chromosomes are more likely to touch each other than across the domain. So you see similar structures both in human, mouse, and in yeast. Okay, so now we have many maps of this coming out, you know, in different species and in the same species with different cell types. But keep in mind that these are, what this is measuring is a physical proximity, right? It doesn't really tell you anything about the function. So if you want to understand how these long distance chromosomal interactions actually function in all these nuclear processes, we actually need a method that can disrupt these interactions, and so we can look at the downstream process, how these change. And ideally, this method should be selective, that when we perturb this, we only perturb the interactions, but nothing else. We want a generic method that can induce interactions at any target sites we want to look at, not only the sites that originally touching each other. We want it to be inducible, so that these kind of interactions can be dynamically controlled. So we came up with a method called chemically induced chromosomal interaction, CICI. So CICI is based on two existing techniques. One is called chemically induced dimerization, CID. So here we're using a classical CID proteins, FKBP12 and FRB. They normally do not interact very strongly with each other, but in the presence of inducer rapamycin, they bind to each other very strongly. Another thing that we used is the fluorescent repressor operator system, FROS. So the idea is to integrate a long array of LexA and TET-O into the genome so that they can recruit a large amount of LexI and TET-R onto these chromosomal loci. So a very common use of FROS is to label LexI with GFP and TET-R with mCherry so that these two locations, chromosome locations, appear as a green and red dot inside the cells. So we can track the movement of these chromosome loci in live cells. So the CICI idea is very simple. You probably already thought of that. It's just to use LexI to bring the two sides of the CID proteins, right, onto two different locations. And hopefully by putting in rapamycin, these two locations will collapse onto each other. And for visualization purpose, we also include LexI GFP and TET-R mCherry so that these two dots are originally appear as separated dots. And when we induce interaction, the two dots should be co-localized. So the idea is very simple, but in reality, this is hard to do because of this issue of free proteins, okay? So imagine a scenario where you have lots of free proteins. Now you add rapamycin, what will happen? Well, the free proteins will bind first. And they work as sequesters, preventing the two chromosome loci from interacting with each other. So for CICI to work, it's absolutely necessary to generate a condition where we still have lots of bound proteins, but not so much of free proteins. So to make a scenario like this, the requirement is that these proteins should have high affinity towards their binding site. If you know anything about LexA and TET-R, you would say that's not a problem. Okay, so in vitro, when people measure the KD of LexA and TET-R, it's on the order of 10 picomolar. Okay, and the yeast nucleus is so small that if you just have one protein inside the nucleus, that amounts to half a nanomolar. So in principle, even if you have one protein inside the nucleus, it should spend most of its time in the bound state. So I told my student, this is not an issue. We just need to control the overall protein. As long as we don't overexpress the protein, we'll be fine. But we got a nasty surprise. We have a method of measuring the KD inside the cells. I'm not going into the details here. But when we actually measure the LexA binding inside the cells, instead of a 10 picomolar KD, we get a 600 nanomolar KD, which is 5 to 6 order of magnitude higher. And this kind of weak binding did not work for CICI. So my student went on a long journey, changing the construct in the cells, genetically perturbed the system. I'm not going into the details, but at the end of the day, we managed to change this curve into this curve. Okay, so we managed to reduce the KD to be less than 10 nanomolar, and this is very important for the implementation of CICI. Okay, so finally, for the CICI to work, another thing we need to decide is where to put the LECO and TET-O array. So before we did anything, we really did not know if any of these methods can work. So we want to put the LECO and TET-O sites into regions that has a high contact frequency to promote the CICI formation. So in yeast, the chromosome conformation is largely dictated by this thing called rebel conformation, where the centromeres cluster at the spindle pole body, and the telomere is on the nuclear membrane, kind of away from the centromere. Because of this conformation, you can imagine the chromosome has to bend very sharply at the centromere, so that if you have two locations that has equal distance to the centromere, they have higher probability of interaction. Okay. So as a proof of principle, we first put our LECO and TET-O both 150 kb from the centromere. And it worked the first time like a charm. So this is in a CICI strain that contain all the fusion proteins I mentioned. You can see without rapamycin, the two dots are mostly physically apart from each other. Adding rapamycin, they come together. This is the histogram of the distance between the two dots. You can see with rapamycin, if we use this as a threshold, the co-localization probability increases from 40 to 80%. In contrast, in a control strain, where we have everything else except the FKBP12 and FRB, except the dimerization protein, you can see the distance did not change at all. So what we're looking at here is not some generic response to rapamycin, it's a true CICI event. Okay. So this is a static picture taken after long-term incubation with rapamycin. We can also do this with a time course. So here what I plot is the co-localization probability as a function of the incubation time. As you can see in the CICI strain, the co-localization rapidly increased after just a few minutes, and in the control strain, it doesn't work. Okay. From this kind of dynamic curve, we can get 3 parameters out. We can get the initial state of things, like the initial distance between the dots, the initial co-localization probability. We can get something that we define as CICI efficiency. Basically, it's the net gain of co-localization after putting in rapamycin. And we can also get the half-time, basically how fast the CICI can form. So now the CICI methodology is clearly working. We expanded our study by labeling LECO and TET-O at different locations in the cell. So the 5 conformation here, the first 4 are intra-chromosomal. That means the 2 dots are at the same chromosome. The last one is inter-chromosomal. We chose these configurations carefully so that if you look at the native contact frequency indicated by the Hi-C measurement before, it spans a large range of the Hi-C signal. Okay. If you just look at the conformation 1, 2, 3, you understand the Hi-C contact frequency goes down simply because of the rebel conformation. The last one, the intrachromosomal interaction, is always less likely than the intrachromosomal interaction. Because if you have a polymer, it's always easier to self-interacting than with some other polymers. This number 4 here is a special case where the 2 dots are put on the 2 sides of nucleolus, the rDNA. And as I showed you before, the Hi-C measurement has shown that the rDNA separates this chromosome into 2 isolated domains. Okay. So basically, we try to induce interactions across this junction. So it would be very interesting to see if we can actually make it work. When we did CICI on all 5 conformations, you can see that the CICI works in every single case. And remember, for each of the curve, we can get these 3 parameters out. Now we can extract these parameters and look at how these numbers correlates with the Hi-C signal. Okay. When we look at the initial distance between the dots or initial co-localization of the dots as a function of the Hi-C signal, you can almost see a perfect correlation. This is not too surprising because if you think about how Hi-C work, Hi-C basically measures a probability for the 2 segments to be ligated together. So effectively, they are measuring a co-localization probability. Okay, so this is not surprising that the co-localization probability fits so well with the Hi-C signal. What is a little bit surprising is when we look at the CC efficiency, that it actually has nothing to do with the Hi-C signal. Even when you try to induce interactions from different domains or has very, very low contact frequency, we can still see a large fraction of dot formation inside the cells. So you can see that for all 5 conformation, the CC efficiency is about 60%. So where does the 60% come from? Why not 100%? So one nice thing about this CC assay is we can actually follow these dots in live cells in real time. So you can literally see the interaction formation. So you can see that in this case, the 2 dots come together, they stay together. Even here, they still stay together. This is because after DNA replication, you actually get 2 pairs of these dots. They still stay together. But somehow, at a later stage of mitosis, these 2 things fall apart. And later on, they can come back together. So this indicates that CC formation is not an irreversible process. Okay. We need more data on this, but our preliminary data indicates that this disruption of the CC interaction is a cell cycle dependent event. So we think that 60% is largely dictated by the cell cycle dynamics. And finally, the halftime of the interaction formation. So you can see that in these 2 cases with very low Hi-C signal, the formation time of CC is actually almost 1 order of magnitude higher than these cases. So overall, so one of them is actually this case. So even though they can come together, it takes forever for them to come together. So overall, this points to a dynamic nature of the Hi-C signal. So if you see 2 dots in different TEDs in different topologically associated domains, it doesn't mean that they cannot see each other. It just means it takes longer for them to encounter. And probably that's the way, so the TED boundary basically imposes a kinetic barrier for the interaction to happen. Okay, I have to go a bit faster. This is just 3 more slides left. So as I motivated you at the beginning, that the purpose of generating CICI is to use this to perturb the chromosome conformation and look at the downstream consequence. So far we have done this one experiment that by looking at the donor preference of yeast mating switch. Okay, so yeast mating switch. So yeast, even though it's a single cell organism, it also has two genders, A and alpha. And the amazing thing is, if you look at a single yeast, it doesn't stay A or stay alpha. It switches from A to alpha to A to alpha every single cell cycle. Okay, and that's why it's called mating type switch. And the switch initiated by cutting the mating site with this endonuclease HO. Remember, at the beginning of the talk, I talked about the HO promoter. That's the HO I was talking about. So this HO would cut the mating locus, and then it needs to be repaired. It can either be repaired by HML, and when that happens, the cells will become an alpha type, or it can be repaired by HMR, then the cell become an A type. But the very interesting thing is, in an A cells, if you have an A cell, it predominantly uses the HML. If you have an alpha cell, it predominantly uses HMR. So that's how the switching happens, right? So this is called the donor preference, and the mechanism of donor preference is still not clear. I think a very reasonable hypothesis is that in A and alpha cells, you have different chromosome 3 conformation. So probably in the A cells, HML is simply very close to mating locus. So when this DNA break is naturally use this as a template and vice versa. However, when people actually use high C to measure the chromosome 3 conformation in A versus alpha cells, they did not see a very significant difference. So for example, this is the interaction between HML to MET in A versus alpha cells. You can see there's a little difference, but not significant. And here, and another pair is this to this is also not very different. So this mechanism still need to be investigated. So with CICI, so this imposes a perfect problem for the CICI, right? Because if in A cells, mating locus is naturally close to HML, then we can put our CICI pair here to force the mating locus to interact with HMR instead. And then we should see a rewire of the donor preference. So I'm going to skip the some details of how exactly we did this. I guess the punchline is here, that in CICI strain, but not in control strain, when we induced CICI with rapamycin, you can see that the usage of HMR increased from just 6% into 27%. So indeed, when we force this interaction and change this conformation, we see a change in the donor preference. And that shows that the conformation indeed plays a role in this process. Okay. All right, so I'm just going to thank all the people that's involved. My lab mostly comprised of graduate students, and I have been very lucky of getting very good, talented graduate students. The first part of the work is mostly done by Chao and Hengye, the two guys here. The last part of the work is a heroic effort from Mandy, this lady right here. And two undergrads, April and Seth, also contribute to this work. And the single molecule experiment is in collaboration with the Poria lab and his graduate student, Ben. Okay, thank you very much, and I'll take questions.
Very nice talk, thank you.
Thanks.
I believe there's a paged version of rapamycin, and I was wondering if you'd given any thought to optochemical control of CICI?
Yes, absolutely. Yeah, that will be the version 2. It's called LICI, light-induced chemical chromosomal interactions. Yeah, right now we actually want to completely get away from rapamycin. The reason for that is in order to use rapamycin, we have to use a rapamycin-resistant yeast strain, and those yeast strains are very sick. So instead of doubling every 90 minutes, these strains doubling every like 3 hours. And we want to use some other set that we don't have to do this. Yeah.
Other questions?
Yeah, people are listening online, so if you give him the microphone, then they will hear the question.
Yeah, if I understand correctly, by the way, it was a very nice talk. This pioneer vector basically intercalate between DNA and the chromosome, and in one hand it hold the DNA, and the other hand it hold the the nucleosome to increase its residence time, right? So then I don't understand how come then that actually accelerate dissociation of the nucleosome, because nucleosome itself is being held by the protein.
Right. So indeed, we find this so-called dissociation compensation mechanism. It's that we find that these factors, in fact, dwell on the nucleosome much longer than it dwells on the naked DNA. So what you said is correct, that it seems to be able to make stronger interactions with the nucleosome than on the DNA. What we don't understand is which part of the nucleosome it's interacting. So because it could make additional interaction with histones, for example, but it could also be that they have stronger interaction with the nucleosomal DNA, which is severely bent. Yeah, that's why we are doing the structural study. We really want to see exactly how these interactions happen at the atomic levels.
May I ask a couple of questions, please? Could we go back a couple of slides to the outcome of the second set of experiments you did? Okay, right there. Sorry, go one forward now. Okay. So you were able to go from essentially 5%, 6% the wrong way to 27% the wrong way, but you weren't able to get close to the 100% that happens in the yeast. So,
right.
So what are your thoughts about that?
Right. First of all, when we induce CICI, it doesn't happen in 100% of the cells. It only happens in 60% of the cells. So that certainly is a factor.
So then double your 27% then approximately. Right. Yeah, fair enough.
And we only did something. So if you look at this diagram, we only did something with HMR. We did not do something with HML. So the proximity of HML is still there. Yeah. It's just a competition. So we only bias the competition towards the HMR side. If we
so maybe 50% actually is pretty close to 100%.
We hope so. Well, if we can do something with the HMR, for example, we can kind of force it to stick to the nuclear envelope. For example,
yeah, you could sequester it somewhere. Yeah, right.
Yeah, exactly.
Yeah. And could we go back to the pictures of the proteins with the leucine, the basic region, helix, leucine zippers?
Okay, well, this would be.
Since I did those, the very first examples of those structures, go forward. Yeah. Keep going. So those are called E-boxes, those CAC GTGs. Virtually all of these proteins bind to E-boxes. I'll make one comment about CBF1 versus FO4. In another system, we found that the leucine zipper can promote higher order interactions among these BHLH zip proteins. So they can form bivalent heterotetramers.
I see. Okay.
So if you look at our paper on upstream stimulatory factor, you'll see all the data that we generated for that.
Okay. Yeah.
And we posited that that could lead to long-range approximation of different pieces of DNA with E-boxes. And so you've got a bivalent heterotetramer, a bivalent homotetramer. So you can bind one E-box and another E-box and bring them together. So that's one point. And then if you just go forward, one more, and one more perhaps, one more. Okay, so the implication is that one of these basic regions binds more effectively to bent DNA. Is that your interpretation? Versus straight DNA.
So the interpretation is this bits of the DNA seems to be, so this bits of the DNA.
So the basic region is responsible for virtually all of the interactions with the nucleic acid.
Yes.
Right.
Yeah.
And the basic regions undergo a so-called disorder to order transition when they hit the right sequence and they fold up in the major groove. Again, if you look at their papers on MAX and upstream stimulatory factor from the 90s, you'll see all this, you'll see all that, and you'll see the biophysics. It was worked out back then. But all those experiments we were doing, of course, were on straight pieces of DNA.
Okay.
Yeah.
Yeah, so obviously these two, even though they are homologs, this bits and this bits,
they're probably not identical in sequence.
They touch DNA in a different way. Because one, like this part of the binding site seems to deal with nucleosome a lot better than this part. Even
so the blue basic region is better at binding to bent DNA.
Yes.
Than the green basic region.
Yeah.
I think this will be very hard to show crystallographically, but I hope you can do it.
Oh, really? Okay.
Yeah.
Well, we can talk.
I hope you can do it. We can talk about more over lunch, but it's intriguing that that it would have a higher, potentially a higher affinity for the for an E-box that's wrapped around in the context of wrapping around an octamer.
Right.
So I actually had a question as well. It's just a quick question. So for K-off rates with and without nucleosome, do you see a correlation between dimers and monomers?
Right.
Because you argued,
right.
Is there enough?
It's not strictly correlated. So in this diagram, so even though this is KD, but the K-on is almost the same. So this difference is mostly due to the K-off. And if you look at, so Reb1 is a monomer that has the slowest K-off. And the other ones are all dimers. And even though they are accelerated to different extent, it still follows the idea that dimers does not bind as well as monomers. Yeah.
But I think you need to be careful when you talk about dimers versus functioning as a dimer versus functioning as a monomer, because though neither FO4 nor CBF1 will work as a monomer, they have to dimerize.
Right.
And then they project the two basic regions within that 6 base pair region. I think to do a really clean experiment here, you would want a dimer that has 2 completely distinct DNA sites. DNA binding site. You would want something that was tethered, that had 2 completely distinct DNA binding sites, as opposed to the site. You see, the CICI GTG is actually one site, and you can't, the BHLH and the BHLH zip proteins don't work unless both halves of the CICI GTG are present.
Right. So in our experiment, in fact, we provide different binding sites for CBF1 and FO4. Different
in what sense?
They both have the CICI GTG, but the neighboring sites is a little bit different. Yeah,
but I think to do a really clean experiment here, you need to separate it being an obligate dimer in order to bind DNA, versus a non-obligate dimer able to bind DNA.
I see.
Yeah. Just to get the cleanest control. That's the point I'm making. Other questions, thoughts? Greg.
Sorry, it's running a bit late.
Yes, is there any change, a difference in the supercoiling topology when a pioneer factor is binding versus something that binds that's not, let's say, the CBF versus FO4? Are you seeing a topological difference downstream in the DNA? And then another, if you have time, semi-related question. Have you looked at, if you talk about binding curved DNA versus, say, uncurved, have you looked at the histones itself? Or can that be getting in the way of something properly binding the nucleosome core?
Right. So the first question is supercoiling. In the in vitro experiment, we let it either bind to mononucleosome or very, very short piece of the DNA. So the supercoiling, any rotational tension will not maintain on the template. In vivo, we have not looked at that. With the second question, it's the interaction, potential interaction with histones, right? And that's so when we saw that Reb1 and CPF1 dissociate much slower on nucleosome than naked DNA, there are two possibilities actually. One is that these proteins make direct interaction with histones, right? Or these factors bind strongly to band DNA. We still don't know which of that choice is correct. Yeah.
So we should probably draw this to a close. Thank you very much, Lou.
Thank you.
Wonderful talk. And before we